I guess since it's the last lesson, we can start as always by taking a nice deep sigh of relief. By now, I think you're experts at taking nice deep sighs of relief. It's really good to do. Remember, we started out with this feedback loop. First thing I showed you, there's the relaxed state. And when you're in the relaxed state, you take deep, full breaths. And then if you take deep, full breaths, you also increase your probability of getting into the relaxed state. The relaxed state is associated with better, take a nice deep sigh of relief, better memory, listening, better curiosity, playfulness, all the things that help us learn and also help us research. That's why I think it's part of the scientific method to take nice deep sighs of relief. You can quote me on that. I'm signing my name. Okay, so today I want to hear from you a lot. Basically, since the last lesson, a special way to review this course and think about open questions that are still there in this field of systems biology that rise up for you. So we'll do this review in a couple of ways. We'll start with talking about the way this course was built and carried out and the exercises, etc. And then we'll talk about what principles we went through in this course. And then we'll talk about open questions. So. Any burning questions before we start? All right. So I talked to some people who came to pick up the exercises, and it came to me that I wanted to appreciate you for sticking with this course, because I know these exercises took some time to do. Several people said 7 hours every week. I don't know if that matches your experience. And so that's a lot. And also doing math is not familiar to everyone. So I was thinking it's really not to be taken for granted for those of you that stuck with this course to the end. I want to thank you because it's always fun to see you. The more the better. All right, so how to do feedback on the course structure. A nice deep sigh of relief. First, maybe I can teach you about feedback in general. So we get feedback. And in science, we don't learn how to give feedback. If any of you ever got a referee report, you know what I'm talking about. How many of you have already got a referee report for your papers? 1, 2, 3, 4, 5, 6. Not everyone, huh? How many people haven't published a paper yet? Oh, about half. Okay, great. Welcome to science. I hope you have a good experience with it. But maybe I can start asking, what is good feedback? What is good feedback? What parts does it have? Useful feedback, I mean. Not, you're great, thank you very much. It doesn't help you. Useful feedback, what does it have? It's constructive. The 3 C's. Constructive. What else? Honest. Okay. Let's call it credible. Which, yeah. When we say honest, what do you actually mean?
Well, you don't want to lie massage.
It's not massaged. It shows you, what to improve, right? Not only you're fine, you're good, and it's also specific, right? What? Okay. So it's the 3 C's. The 3 C's: constructive, credible, concrete, and concise. But in particular, we're going to practice it in a second. What to keep and what to improve. So we call this the feedback pita. You want to be the purpose of the pita. The purpose of it is to help the other person. It's not to show where they're wrong. Right? And inside the pita, there's 2 parts: what to keep and what to improve, called plus delta. And they're specific, something specific. It's not like you're a good student. That's not specific. It's like when you found that software that arranges all your software pieces and makes your master's software so much more efficient. I was very impressed. Daphne, for example. Okay. So that's specific, right? That's what to keep. And what to improve, also specific. You don't say, what is this? Any second grader can run a better gel than this. What is this? Right? There's no control lane. You say, oh, good, we see a band. That means the antibody worked. Now, do it again, but this time add a control so we can see if it's actually the drug that had this effect. So what to keep, what to improve, right? You found the right concentration of the antibody. You have a band. Okay, so we're going to do that on the course. We're going to practice it, okay? So I'm going to write here plus. Then we'll do the delta. So I'm now talking about the structure of this course. I want to learn from you. What helped you learn? So my question is here, specifically, and then you get to tell me what to improve, okay? What helped you learn? All right. Go for it. Huh? When I explain, can you be more specific? Huh? Like explaining the feedback? Explain what is feedback?
No. What do you mean?
Can I explain? Yeah, I'd like you to start telling me things about the way this course was structured, the way this course was run, what I taught, the way I taught it, the exercises. We have here Pablo and Jean, who were running the tutorials, right? How many of you went to the tutorials? Almost everyone. Wow. Okay. All right. So next time I do the course, what should I keep?
The exercise.
Yeah. What about them?
Is it good? They're good in reinforcement.
What?
They're good for reinforcement. Exercise.
Okay. You can just say. You don't have to wait for me. Okay. Okay. What else? Don't be shy. Yeah. How many people watched it? awesome. Okay, thank you.
I would keep the chalkbook.
Chalk format, not PowerPoint. Why?
Because when you see an equation in the PowerPoint slide, you look at it, it's hard. You don't develop it. You have time to see it.
It's time to see how equations develop. Yeah.
Personally, I disagree with this point. I personally prefer PowerPoint because then you have all the material written down for you for later to go over.
PowerPoint.
You can put the development of the equation in the slide.
Arranges the material. Okay, so I'm breaking the rules because soon we'll get to delta. But let's keep on with plus. Because we'll get to delta, don't worry.
Having the book, to review things you want to see again.
So, I'm telling you, I don't know if I told you, I'm writing a second edition. This course is helping me a lot, so some of the lectures here are not in the book, but they will be. And okay, thank you. For example, to say this lecture will be chapter 9, 11.7. Good. I'm going to use it. It's something I'm not wasting our time here. I'm going to use it. This is for the benefit of the next class. Also for you guys to practice, because we don't usually do this in science. Reflection. You're going to teach one day. When you teach, I hope you can gain from this course a little bit on reflection on ways to teach, not to teach. Yeah.
Just something.
Yeah, of course.
So what had me learn was that every class was structured like a story. Like there was a consistent logical development, like in a book, like in a novel.
Okay, thank you.
Each class had a clear message. It was modular also. Class 3 didn't depend necessarily on class 1.
Right.
Still connected.
Right, that's right. Yeah. What about this side of the room? What pluses did you perceive up there?
Regarding questions, there's not always time for questions, but when a question is asked, it was always taken into account, fielded. You made sure that you understand what the question is and how to answer it.
Okay, so I try to make sure that I understand what the question is. So I want to just teach you something. How many have given a talk yet in science? How many have not yet given a talk? Okay, a few of you. Okay. So both of you that have, those that haven't, and those that gave 100 talks already. After I gave 500 talks, I finally learned how to respond to questions in my talk. Because for me, the question period was always very stressful, because I was thinking, maybe to get it over with as quickly as possible, because people will find out that, give me some criticism, I don't want to hear it. I was stressed. And so somebody would start asking a question, after three words I would start answering, because I would imagine I know the question. Do you know the feeling? Have you ever done that? Like answer the question before you...
No, I always listen to the question, understand it, and then...
Good. I wasn't like that. So I learned some technique. It's a 5. I make eye contact. I listen to the question to the end. Then I repeat the question in the asker's own words, if I can. And then I look again to see if I got it, because if the person is like this, I say, okay, can you give me more context? And then I answer the question, and then I ask, did I answer your question? Now this sounds very technical, but it's a framework that really relaxes me, because I know what to do. And it gives me a lot of time to think. So I try to use this in the course. Sometimes it works better, sometimes worse. You could notice sometimes I just don't understand. I'm sure you noticed that sometimes I answer without really haven't understood the question, despite all of this, because it's really difficult sometimes to understand questions. And so that helped me a lot. What else? Yeah.
In addition to that each class was structured like a story, there was also a connection between classes. And there was, for example, the recurring example of the lag Z.
Oh, yeah.
Overall, so it's a nice connection throughout the course.
Okay, so like e.g. lack z kept coming back again and again. Right?
And the motifs that we
Yeah. Good. You see, some things, yeah.
I like the more interactive parts where you ask the question, you try to solve it together.
Okay.
Even when we voted again after consulting.
Okay, so yeah, so this the dyads, what's called the dyads, which are made of asking a question, vote, then a partner, revote, and then ask someone to solve on blackboard, on whiteboard, right? Remember, it has these parts. So I don't know. It's an experiment for me. So I'm just asking. Please be honest. How many of you would vote to keep it? Okay, that's good. It's a majority. That's fine. Okay, so I was unsure. I mean, I didn't invent it. I learned it from. As I told you, there's a paper about this. And okay, what else? Okay, maybe we move to the delta, to references to the book. What else can I do to improve? Or we do also in the exercises?
I think that the exercises were more oriented at testing our knowledge and not helping us learn what we just learned.
Make exercises oriented to help us learn, not test our knowledge.
Yes, maybe
Yeah.
maybe the fact that it's 50% of the grade doesn't help. I mean, if it's like 20% of the grade, then
Make it 20% of the grade.
yeah, because then
Why? Definitely not.
But then-
Wait, let's say we can hear different deltas, and you can let's
say
listen.
why.
It's not. It's okay to have different opinions. Yeah.
I'll say why also, because also with TA, because then you can, if student comes with questions, then you can really try to teach them what they want to know and not really be concerned about testing them and the grades and the distributions or whatever. I
So
think this is the purpose
I want to
of
understand.
exercise.
You're saying this is for the one-on-one interaction with the TA, or yeah. So TA can supply additional teaching.
Yes, and not be more
I see, because if it's a big part of the grade, it's more like I can't really to give you the answer. If it's a small part of the grade, you can.
yeah, you can really
Understand,
make sure that
I
the student
understand.
understands
Got
and
it,
then
okay.
test them at the end.
Yeah.
So if you put them in a small percentage of the grade, the
Not-
motivation might not
that
be there.
don't get hung on the
Okay.
20
But
whatever.
I don't want to make it a discussion. I just want there's a trade-off, always in many things, like we saw in the Pareto front. There's different tasks for the exercises, and I was thinking, what's the purpose of the exercise? What's the purpose of an exam? So the exercises have purposes, like you say, at least two. One is to evaluate, and the other one is, of course, having the exercise of each week makes you want to review the material, right, or to work to exercises, and then. So there's a bit where your ethics are as a teacher, or others would say, the weight you give to the different values, and this is, by the way, there was a TED talk about this recently, how you make hard decisions in life. Hard decisions in life, like the hard decisions, the problem is that they have two values collide. If one choice was better and on all values, you would make it. But you're stuck because two values collide. And then, as a human being, you have to just decide what's more important for you: living in the city or living in nature, for example, whatever. Living in Israel or living abroad, whatever. And then you define yourself by making these hard choices. So, in the past, I made really, really easy exercises. And not satisfied. So this year it's more on the. So okay, but let's continue, please. More. I'm saying this is a real issue to think about. Yeah.
In a few cases, when equations were drawn, the voltage, I think, explained during class, and only in the tutorial it was explained, but at that point, it was hard to follow in the class, I think.
Tell me, how can I improve this class for next year? Yeah.
I think that the exercises were much
harder to solve unless you actually went to the tutorial. Like they were more connected to the tutorial than to the actual class.
Okay. So it's like the two exercises. And are you saying exercises solvable just based on what was in class?
Not necessarily just on what was in class, but more connected to what was in class.
Okay. Good.
Connected to the modular connected
Yeah.
that I think that in some cases it was kind of hard to understand that you started a new topic and you should have make all the topics saying this is a new subject, like when you start with the Tarita font and only later on you connected it to the circuits or
Right. So,
I want to say that the counter thing, that not to this one, but for people who don't have background in biology, we also sometimes had trouble following you when you were using more like.
So what would you advise me to improve, for example?
So more like help for.
Right.
More. So maybe more of that if you don't have time in your show.
More tutorial.
Or maybe for both, if both sides feel like.
Right.
similar suggestion in 2 years ago there was a course of Nirgov and Michael Elbaum and others, and we had separate tutorials for the physicists and for the biologists.
Yeah. Yeah.
Also as a non-biologist, I think it would be helpful if we get like a list of things that you need, like background, like biology background, and like read on your own.
Good.
Because I personally didn't know what I didn't know.
That's really helpful. For example, maybe let's take a nice deep sigh really for the way we don't forget you. For example, if we put a list of Wikipedia terms or something, transcription, translation, natural selection, mutation, recombination. Yeah.
Exercise handout sheet.
Yeah.
Recently, right? So I observed that there was quite a high dropout rate from the course, about 30%.
Right.
So it would be nice to look at the dropouts from the class and see in which field they belong to. So we might be able to cater the class to make it more understandable. Is it more biologists or more non-biologists who drop out from the class? And we know where
Good. Yeah. Mm-hmm. Thank you. Yeah.
Maybe it would be, nice to have the solutions for the exercise, like tutorial answer.
In hard copy or?
This is data sessions. I wonder if I can ask a class about like the office hours were not so successful.
Yeah. So you said the office hours were not successful in what way?
We didn't have a big turnout. It was like 1 or 2 persons per week.
Some people used the office hours and others didn't, and you'd like to know why?
Yeah.
Or how to improve it?
Yeah, next time.
Does anybody have feedback for Jean on office hours? If there's anything you think, yeah.
Yeah, only just after the tutorials. And you should see a much better uptake.
Day before. Yeah. So you say find problems and exercises. This is in the office hours. So the office hours helped us understand where the exercises could be improved. Right? Again, since if you study electromagnetics or if you study bacterial genetics, these are like fields existing for more than a few years, maybe 100 years. So there's a lot of nice exercises. Here we're right at the frontier of science, so we have to invent exercises. Inventing exercises is a very interesting problem, and actually, it really tests your understanding. And we learned a lot from you about the exercises. All right. So we got. I see that you wrote it, right? Yeah. Okay. So this is about the course structure. And now, it's about the course structure and also about a way to give feedback. Oh, I forgot to say, what do you do as the person who got feedback, right? If you may know this thing in Israel, if you get a compliment, you say no, or like you say great cake. It was just by chance or mistake, or it wasn't me. It was no, because it's my aunt take a breath, deep breath. You see, it's in my family, and say thank you. Now, what do you do when you get plus and delta? You take a deep breath and you say to keep, right? That's your autonomy. You don't need to argue, explain yourself. You just say thank you. We wrote it down. So that's the end. That's the end of the feedback pita. This is what to keep. Unlike a real pita, where you can't always choose what to keep because you've already ate it. And then the person who wrote the paper, the first author, goes to them and listens to them to get their plus and delta, and writes it down, and doesn't explain to them what they meant, because the reader of the paper won't have that benefit, right? You won't have the author next to you to ask, prove the paper. We take what we need, and then the other half reads it and gives feedback in this way. So the rule is, for the person who's the first author who's getting the feedback, is there's magnets on your ears that open their ears like this, and there's magnets on your lips that close your lips like this. Just listen. Write it down, and don't argue. Okay, so I guess this meeting is also spiced with this topic that I love: how to actually be a scientist. So if you answer questions, all these are like really important things for being a scientist that I think most of us don't learn. But at Weizmann we're going to change that. We're planning to have workshops, modular workshops for graduate students, postdocs, how to deal with difficult personalities, how to lead when you're not in charge, right? How to take a pause. Okay, now we're going to look at the principles. So this is again reflecting on what we learned and in the way that it will stay with you, have a bigger chance to stay with you. And I'm going to go. Each one is now going to say something. Each one is going to say something, and each one is going to go like this. And I'd like you to think of one now that you thought, wow, this is cool, interesting, new, and it should be a specific moment. It could be a small moment, okay, in your personal moment. You said, wow, this is interesting, this is cool, this is new for me, or something I see in a new way, or okay, and we're going to collect them. And from you, all right? And we go, and please say your name also, and what is it? Okay.
I'm Yossi. The moment is the modularity paradox.
Can you just say what do you mean by modularity paradox?
That the circuit involves much faster when you change the goal.
Okay. Evolution is speeded by modularly varying goals.
Ken?
Okay, just say it.
My name is Ken.
Yeah.
I think the Pareto problem.
Can you just make a full sentence?
I think that was a very interesting concept to see the trade-off.
Visualize the trade-offs. Yeah.
Liran. I like
Pareto front applied to gene expression in cells? It's okay. You can say it again. Say it in your words, though. By the way, you can repeat, right? Like,
it's surprising that when you change the code, the evolution like
okay, great. Could we go to you, please? What's your name? Yeah. You remember that lecture? Fitness cost and measure it.
I actually, one of the very first talks when you started describing the feed-forward loop and the different gates and then data, and we saw how well it fits, and then how we can predict what type of regulation based on the model itself.
Great.
Very nice.
Okay, that was a good moment. Yeah. E-L-I? L-E? Is this right? Okay.
The Pareto front, because I think it's sort of amazing how evolution makes very complex traits fall into like a specific line or space of values just because of the constraints of evolution.
Due to. Yeah. Right. So. Kinetic proofreading and conformational proofreading, if you remember these two. DU.
I really like Pareto fronts, especially because they make you look differently at things. For instance, are short distance runners and marathon runners different specialists?
BG. Okay, so
medium distance runners,
sprinters and marathon runners
exactly,
as archetypes. Great. Okay. So maybe, Jana, I could ask you to do a snapshot of this with an iPhone, or Pablo, after we finish the board, and then I'll. Okay, Daphne. Okay.
Activation and repressor are not symmetrically the same.
Right. Yeah.
Lily?
L-I-H-I?
LWHW.
Okay.
And I like the evolution, the speed of evolution by modularity. It's really fascinating how you can modulate.
Give the needs and the environment, and vice versa, the cost and the value.
Yes?
Ori?
You say it?
Ori?
O-R-I?
Yeah. And I like the part about modularity and how it evolves as a result of modular inputs.
Omer. I'm also going for the Pareto front.
Okay. It's actually very useful to see if there's repeats. It's good to get a sense of. Yeah, go on.
What I like is the realization that what we're seeing is the projection of the organisms into a trade space, and the truth that we're seeing gives you a lot of insight.
Traits affect what we see, or the trait choice. You said, right? Yeah. And now we continue like this.
My AYN.
Okay.
I was surprised by how quickly entire units or circuits of the cell can be shut off by not being required anymore.
The circuits could be lost in evolution. Yeah. Yeah, right?
I think the first class has been regulation. When I saw the simplicity and the elegance of the first equation, it was really nice to see how to simplify.
Simplify gene regulation. Great.
DNA changes, how you take a very complicated problem and you have a very simple rule how to find the solution.
And simplifies the complex DNA changes and biology. Yeah?
Also for me it was interesting because it was connected to something completely different. Like I only searched a completely different topic. So it was
Can you just give a title to that topic? Statistical physics.
energy landscape.
Energy landscape. Yeah.
Changing goals can speed up.
You don't have to say it out loud. Yeah, go ahead. Go ahead.
Changing goals can speed up evolution.
Speed Up by MVG.
The question of whether the extras get genetically constant, and the question in the exercise shows it can be reversible, so it can answer this.
And if it's reversible, this is not energetically costly, there's no kinetic proofreading. Okay, great. So let's photograph this. It's nice to relive the moments that you're mentioning. Of course, as a teacher, I also feel some moments very excited that we're together. And some moments it's more... didn't work. Okay, so I'm going to keep on going from here. Okay. Let's go on. Where are we? Can you spell it? Cool. Okay.
That's because I never accept why some genes are regulated by activators and others by repressors, and I really like the answer.
Keep your promoter covered most of the time.
Kind of like the part about the bird. I never thought about the legs of our bones and the legs and hands.
Burrito. Okay. And now we go like this. Couldn't hear exactly what you said. So the fact that we could reduce, you said? Deduce? I said we can deduce the tasks from the... Is that what you mean? Or and multidimensional trait. I think I didn't understand exactly. Could you explain to me just again? The moment of insight was?
Just that very different we see is not all the dimensions that exist.
Got it. The Bruin front. Okay. Can you spell? Can you spell it? I.
Again.
I. Like this?
Hey.
Like this? Okay. Thank you. HADAS.
I liked a lot of things people already said.
Of course.
I'll say a new one.
Yeah.
Integral feedback is integrated in chemotaxis.
Okay.
Deal. So I like in the tutorial that Hila gave, she talked about research about cancer cells that Elon and triangular quite found.
So this is mentioning that cancer cell gene expression falls in the tetrahedron.
Triangle. And that you can infer from the corners.
Thanks.
One.
One.
Right now we show using the shape of graphs. The last lesson about the two parts. One is it's like the line when it crosses.
Okay, so inferring properties of circuits just from the qualitative shape of the graphs. And the examples are this what we talked about, the homeostasis in cell circuits, right? And bistability, right? Okay, it's bistability. And also, you mentioned the negative auto-regulation. Did you? I don't remember. Remember where we just say, okay, if the graph, if it's, if it goes like looking at just distances. That's a new thing I added to the course. I also like it. Okay, so that's the one. We go to.
Are you?
Yeah.
We're looking at the immune system, the T cells, and how the receptors constantly mutate. They're willing to throw away 98% of them, just so you'll be able to attack lots of different kinds of proteins.
And that this, so that they won't react to your own body, for example, and they'll be. Okay, continue. We are almost finished. We have 1, 2, 3, 4, 5, 6 more. Yeah. Yeah.
I really like the general principle of looking at biological systems in terms of trade-off between these two functions. I think it was, throughout the course, not only on very fun classes.
Right.
My name is, and I have to say that what really surprised me about the course was like how biological systems could be collapsed. I mean, it's so complicated, they could be collapsed into simple models.
Complex biosystems.
Yeah. So as an example, I took a class in this chemotaxis by Michael Eisenbach, and I remember thinking, this system is so well characterized. Why isn't anyone doing anything quantitative about it? And then I came to this class and I realized this idea was like 10 years too late.
Yes. Chemotaxis system, so well characterized. It was one of the few places where you can do models very effectively. Okay.
Ilya.
Spelled?
ILYA. And I like the concept of robustness in biological circuits. saw that there is a design where the output will be independent of the concentrations of the proteins involved in the circuit, with the two-component bacterial system as an example.
Great. So remember that class? The two-component system with the kinase, it's also phosphatase and phosphotransfer, all these details working together to give you a robust input-output relationship. Pablo? This one?
Well, this one I like to put the faces in play.
Yeah.
Really.
Okay. Yeah, that's nice, right? Faces to know all your names. Yeah.
But no, last lecture, the paradoxical, the one signal does the two opposite things in the cell. It doesn't make sense. It still wonders me though.
Jean?
For me, a big moment in the class was to see how we could use the phase space analysis to understand the function of complex circuits, like oscillators, autoregulation.
To understand oscillators. I wish I understand oscillators, it's still there. And the negative autoregulation. Yeah. I admit, flowed for me. It was a lecture that really flowed for me. And I went and wrote down a chapter in the book right away. Front lecture. That I could explain the Pareto front concept in an hour and a half that was in a way that was satisfying. See that for some of you this was also a highlight. And also nice to see that almost every class is represented here. I know that you also made an effort to do that, but is there something here that's not here? I mean, I'm not saying that it's not good material, but is there, because I just asked you about the highlight. It's one thing, right, out of many. I'm sure you had asked about applying the Pareto front to circuits, and then the minimal walk. You remember you did the game with the pentose phosphate pathway, which was way a big highlight for me, that you found another solution. So, minimal walks in metabolism. What else is can we say? Fold change detection. The possibility of detecting relative rather than absolute changes, and circuits that can do that. What else? Oh yeah, so we had a class about all motifs. We had a class about negative regulation, positive regulation, then a class about feed-forward loops, single input modules to make temporal order and dense overlapping regulons. Temporal orders. And so, thinking about this course, I'm thinking we divided it into parts, and the parts had. My intention was that you experience the way of thinking in biology, of just like you say, you can say collapsing complicated systems. It's clear the biology is right now the most complex object that we study in science. It's more complex than a computer. And it's more complex than a society and the level that we study it right now. We can study down to the atoms inside the ribosome that's inside the nucleus of a neuron that's inside the amygdala in the brain. All these levels. And still there's a hierarchy of fictions, they call it, which is stories that we tell ourselves with math, that goes from the extremely detailed, where we try to add all the rate constants of all the proteins we found and get hundreds of equations with hundreds of unknown parameters, and then simplifying up to the level of toy model, where we end up with one equation or two equations or something we can solve analytically. But the art is to, and then the thing is, you can torture biological material, do experiments that force the biological material to be in a regime where that model actually applies to the reality. And why is science so successful at producing iPhones and stuff? Is because the ability to do that, to torture material into places where concepts can apply is what engineering is. Because engineering is taking material, taking materials like all the silicon and tellurium oxide and stuff. You can make very complex mixtures, but if you make the metal line straight and the silicon, torture the material so it fits into our mathematical models, you get an iPhone, basically. So that's what's going to happen to biology in this century. It's happening already, but turn into an engineering discipline where we build new people. For better or worse. And it's always better, let's say, if you can get rid of the necessary to have root canals, for example. Why not? And from a scientific point of view, the miracle that you can actually make principles understand, in my opinion, makes biology right now the most satisfying science ever. Because evolution, for evolvability and stuff, to human beings, for instance, you can't understand a network with 1,000 nonlinear interactions, a generic network. It's impossible. But if evolution worked on it, and it becomes like a modular, and that module is made of smaller modules, and those modules are made of just a few network motifs. Those network motifs are made of their parameters lie on a Pareto, one-dimensional Pareto front. You can get to the end of it, the bottom of it. Inside the human body, each one made of hundreds of subsystems. There's a whole century of work to understand important problems. How does the kidney work? How does the immune system work? I'm not even talking about how the brain works, of course, which is never level. But so a lot of understanding of ourselves, the living world, in a very satisfying way. We're just at the beginning, and I find that very optimistic. I think there'll be a century where many people are going to be able to do important and satisfying research. It's not going to end very quickly. And also, if we think about the overarching principles that we talked about, like the reuse of a small set of circuits, modularity, robustness, which means circuits are designed to withstand the natural insults that biological material is heir to. Principles of error correction. We talked about paradoxical components and all this inside robustness and optimality, right? The idea that you can. There's hope that you can define the constraints and the multiple objectives in a way that will make you make sense of why this parameter is 2 nanomolars and that parameter is 2 picomolars per second, etc. So optimality is great for us to. I believe that if it's just the tip of the iceberg. There's many more principles. I think we have the minority of principles. The majority is still to come, and that's also tremendously exciting for me. And I can say I don't need to retire. I can still have a couple of decades of good research, because what I'm interested in is those principles. That's my srita. All right. So there's also a whole range of principles that Namabarkai is working on about pattern formation, how to make precise patterns and scaled patterns that I was considering adding to the course, but I didn't in the end. So there's a chapter in the book about that, and there's a lot that happened in the last few years. So let's turn to open questions, okay? All right. So your fold change detection apparatus noticed that the beats per second went up. Nice deep sigh of relief. Shalom. As we had a song from the 80s. That's great. Okay. There was a question about the exam. Maybe we can say some parameters about the exam. Jean, would you like to help me?
Yes. So the exam, probably, it's going to be on the 3rd of July.
4th of July?
3rd. In the Weissman Auditorium, physics building. The format right now, it's going to be a mixture of multiple choice questions and open exercises. Short exercises and
closed material.
Closed book, yeah, closed internet. there was a specific questions about whether we will allow formulas or like 1 or 2 sheets with some more formulas, and something we have to discuss.
No formula sheet.
No formula sheet. Problem solved.
No formula sheet. Okay.
So that's pretty much it.
More? Any questions?
Are the questions going to be like the exercises?
But first, let's take a nice deep sigh of relief. Look at this. I wish I'd do sandwiches, man. But soon, soon. The question is, will the
multiple choice questions. So in that sense, they won't be like the exercises. And also, we don't want to give questions that were already given in the exercises. But it's going to be comparable, right?
Yeah.
It's, because also it's the same.
Jean, would you say that if people understood the exercises,
Yes.
they'll be able to have an easy time in the exam?
That's what we want to, yeah.
That's our goal.
That's our goal.
Is that right?
Yeah.
Most of the exercise will practically take it in.
So the comment is that most of the exercises.
Without tutorials.
That's right, but now you've done all these exercises, so you're in good position to and of course, you know, the exam we're going to test it, so make sure that it's doable. So, but I get the comment that so maybe we need to consider, you know, if we have to analyze some circuit that remind of the topology of the circuit. So anyway, we will try it out before, so to make sure it's doable. I mean, our goal is not to, you know, our goal is not to get everybody to get a.
How many questions are there?
The contents of that question.
All right.
Will the questions mainly be about like the scientific principle? In the exercise, mostly the information you needed about the system was in the question already, or do we have to remember the various systems that there were and like compare between them?
So if you ask me, I think I don't want to you have to memorize arbitrary logical details, but it's good you have to memorize the principles and you have to know the principles that you learned about in class. Right? Would you agree with that?
I would agree. Try to make the questions contain the information that you need. Our goal, yeah. So we'll be around in the exercise, in the exam. I'll be around. I think you guys will be around.
We'll be around.
We'll be open to listening. If something's not understandable, we'll change. We'll write on the blackboard to give more information. So if you don't understand something, you call us, and we make sure that everybody gets that information. What time of the day? Do you know?
10 to 1.
Okay, 10 to 1. Also, I don't like to test people for speed, so we'll try to make an exam that we're supposed to be able to solve in, half of the time. Some people go slower, especially under stressful situations. Right? We said about this feedback loop. When you're stressed, you don't have your deep, slow, full breathing. You have your fast, shallow breathing. I know what it feels like. You perspire, your hands become itchy. You can't concentrate. So that's not the point, to see if you can go fast. So we take that into account.
Will there be like a choice of questions, or will it be like a?
Yeah, I don't know. We haven't written it yet, so we haven't decided yet. So okay, let's go on. Okay, I know there's a lot of open questions about the exam, but let's go to open questions in systems biology. Okay, I'm gonna erase your. So I actually found that the winner here is Pareto front 8, and then there's a second reading. Second is proof is MVG is evolution, MVG modularity, MVG modularly varying goal, and then proofreading. And I'm kind of happy that Pareto and proofreading and modular varying goals are both new chapters for the second edition. So definitely gonna. I even wrote them already after the lectures because those were really flowing lectures. I wrote them on the plane, Stockholm, without my kids. Nobody needed anything from me. I didn't need to make sandwiches or anything. All right. Open questions. And right, so we have circuits. So one vast area that's open is understanding cells. Cells communicate by secreted signals, like in the immune system. And they communicate by cell-cell contact. And they can communicate by, if they're a neuron, by electrical signals. And each cell itself is like we saw, it itself is like a computer with its circuits inside of it. And now, vast systems like the immune system, like I said, like the brain, all the organs in our body, the hormones which the brain secretes and affects the cells in our body, and they secrete back. All these kind of feedback loops. All these are wide open systems. Especially some of the tasks that they do, like how you recognize self versus non-self, how you avoid damage to the body, how you remember past pathogens, how you know where in the body to go. All these things, all these kind of computations. The brain, all the questions of set of courses working about that. Why our organs are built the way they are. For example, has this hexagonal arrays of cells, like B'chat HaMeshushim, with the blood vessel going in like this, and blood vessels taking out like this, and the cells do their different functions, apparently with relation to their position between these veins. And the liver has multiple tasks. It needs to make glucose. It needs to make blood clotting factors. It needs to make albumin. It needs to make many, many things. It's like the metabolic hub of the body. And how do the cells divide the labor between them? And how does this relate to their organization? All these things are open, you can say. And technology is also coming to a point where you can address this, especially the ability to look at single cell gene expression, and this cell and this cell. There's a lab here who's pioneering this Shalevitzkovitz lab, addressing questions like this. Also, inside our body, there's this area living inside us, and they interact, like communicating cell ecologies. So understanding that is also in the frontier. Labs like Iran Segal is looking at this now. Immune system, Neil Friedman lab. This is all for you guys if you're interested in ever exploring topics that are in the frontier. These labs are excellent labs at Weizmann, right? Working on systems biology. Or the problem of egg to embryo. How to make robust patterns. Cells produce new materials. On Milo lab. So we have a lot of the frontier of systems biology. This is probably one of the most central places, if not the best place to do that right now on Earth. You can go to other planets, but that's a big relocation step. If you want to stay here, you're lucky because we have this amazing group of systems biologists. There's more, not only these groups. That are, I think all of these people came from outside of biology. Nir is a physicist, and we actually studied together the first physics, and Iran came from computer science. Shalev came from physics and engineering. So that's a recurring theme. Even though I would say that it's a transient. In the end, in my view, all of biology will be systems biology. What do I mean? It's not that biology will stop it. We'll integrate the findings inside a mathematical view, just like in physics. I think that's the future of biology. So systems biology will become, all biology will be systems biology, which is a molecule or interaction together with a prose story about how it relates to function. But it will be that plus putting the information inside a mathematical model to help it interface with the rest of the knowledge on the cells and with general principles, etc. So that will be the bottom line of biology. And then this kind of course will be the biologists here. Biologists are trained to think this way. And but right now, because of historical reasons, these people come from physics and computer science, and there's still room for people from physics and computer science, plenty of room to join us here in biology. Did I forget any lab here? Maybe somebody can help me. Do you want to mention? Okay. Iran Elinav. He's working in immunology. He's a MD, right?
Yeah.
Thank you. The problem, I mentioned one problem that I find fascinating is reverse ecology, which means understanding the environment in which an organism evolved based on its circuitry. We saw this in the demand rule, that we could see if it's an activator or repressor, we could guess whether the signal is common or rare. Remember that in the environment? So that's a kind of step towards reverse ecology. But for this microbiome living in our gut with all these species communicating and secreting different things, what's going on inside our body in terms of that environment, that special environment? From a bacteria point of view, once bacteria evolved, when was it? 3 billion years ago? I don't remember that number exactly. 3 billion years ago. There were big changes, like there's suddenly more oxygen because of cyanobacteria, etc. The fact that there's multicellular organisms is not very impressive for bacteria, except that we're very nice fermenters. Or in other words, bacteria, you can say, domesticated us very successfully to carry them around and supply them with nutrients. So bacteria are very, very successful in terms of the number that there are. So in our body, I'm sure many of you know this, there's more bacterial cells than human cells, and there's a big factor. Does everybody know? Is it like factor 100, 10, 1,000? 10? 10 times more bacteria than cells in our body than human cells, right? What? Even though it's just a few kilos, they're much smaller than our cells, right? So if it's so if it's like 1 kilo of bacteria and we're 100 kilos, but our cells are 1,000 times bigger. Okay, makes sense. So they have 10 times more bacteria. Each bacteria has they have more. There's more bacterial genes in our body than our genes too. Maybe something. Many more species. So there's, I think, in terms of genes, there's probably 100 times more bacterial genes than human genes in our body, right? So in terms of what they can do, like metabolically, I think when we think about the levels of understanding, I said there's a hierarchy of fictions, right? So if we do the Pareto front of and the understandability.
How do you define what is our genes and what is bacteria?
Oh, so
the bacteria is not part of our body?
yeah. So if you look at if you take a cell from your body and sequence it, you'll find 3 billion, a little bit more than 20,000 genes. And then you look in the gut. So topologically, right, we're like a donut. We have this tube, and topologically, the bacteria are outside our body, even though they're inside. Right? Like a donut. So it's okay. So let's consider them outside. They're not part of our body. They're inside us, but and now you sequence them, and you find out that there's thousands of species. E. coli, for example. This coli comes from colon. Colon is a mea gas. That's where it comes from. That is its shady past. But that's just one species. By the way, part of its definition is its ability to grow on lactose. So it's the species that can eat lactose. It has lac genes. The other species, most of them don't have it. But there's thousands of species. If I'm not mistaken, correct me if anybody knows it. Right? There's thousands of species in the gut? 500? Okay, order. And each one of them has its own several thousand genes, which are different from our genes. So if you sum that together, let's say there's 1,000 species, each one with 1,000 genes. Let's say it's 1,000,000 genes compared to our 20,000. So that gives us a lot of extra enzymatic breakdown the plant fibers that we can't break down. Or for cows, they can even break down cellulose so they can eat grass. We can't do that, but they can do that because of their bacteria. They're mammals just like us. They don't have cellulose. So there's, just to understand our bodies, we also need to take into account this networks of genomes interacting down there.
I think it's also worth to mention in the context of reverse ecology, also viruses are adapted to live in our bodies as well as cells.
Thank you.
Some of the mechanisms are very sophisticated.
Let's talk a little bit about viruses. I'm personally very interested, and we're going to start a project in a few months looking at how 1,000 proteins in space and time respond to a virus attack, to influenza attack on a human cell.
So this is another point of view, but also how the genomes of these viruses
To deal with us. Yeah. So viruses is fascinating because they're tiny, right? The viruses, the E. coli phages are like the smallest thing you might call. They look like, this is the T4 phage, I think. It looks like a lunar landing apparatus. It's 10 nanomolars long. It has this kind of dodecahedron. It's amazing. And it has these legs, and it lands on E. coli and contacts its entry protein and injects its 10,000 bases of DNA into E. coli. Integrates into E. coli's genome and stays quiet unless E. coli is stressed. When E. coli is stressed, it wakes up and starts taking over, hijacking its entire translation machinery and making 100 copies of itself that burst the cell and infect. That's called the phage. Because let's say some phages have only 4 genes. 4 genes. Okay, they're not exactly living because they need the E. coli in order to reproduce. And then the viruses that are in our body and that attack us are not much more complex. They usually look like this, right? They look like World War II marine mines. And they also have, they're very good at mutating. So their mutation rate is very high. It could be, let's say, 10 to the minus 5 per base per generation. And they really know how to hijack our bodies. So P53, the guardian of the genome, that's how P53 was discovered, for example. So understanding this, and of course with the threat that we'll all be wiped out by a big plague sometime. It's on my mind. Of all the threats, I'm scared of that. I don't know why. Each person is scared of something else. But. So that's another whole ecology, right, to understand. And it's alluring because they're simple in terms of number of genes. But actually, when you look at them, a lot of them are non-modular. I wanted to tell you that. A lot of these phages, which have a lot of selection pressure to have minimal genomes, they start using dirty tricks on their DNA to make it to make them less understandable for us. For example, what do? They have a coding region. This is a gene, so the start codon and the end. And here they put in another gene. And here they put in another gene. And here, what does that mean? It means that the genetic code here is encoding some amino acids, and the genetic code here, the same sequence is used in two different ways. It's like solving a very difficult puzzle. How you make the same codons translate one amino acids in one way and use the same genetic code to translate amino acids in another way. So they start overlapping genes, overlapping regulations, and using all the dirty tricks of like once I'm transcribing this gene, the DNA is uncoiling, and that affects how much I'm transcribing that gene. So it's difficult to on the modular, non-modular spectrum, they're pushed towards the optimal non-modular side sometimes. And so that's. But we have these different models, and we have very simple toy models here and full equation models. There's a Pareto front somehow, and in this course we were trying to be in this region, and I chose systems actually, where you can make understandable models that are also very accurate. But if you want to understand, let's say, the whole body, the whole liver, etc., how will it work? So I'm thinking that maybe it'll work like how we understand this. I don't think there's a human being that understands everything about the iPhone, right? But there's people who can understand the process of making this transparent screen, for example. And they have people who can, work the machines, and all they need to do is to be able to communicate through interfaces with the people who want to design this button here. Understanding of the modules, and then you need experts on each module, and the interfaces have to work right. So that's how I foresee it will be very accurate, but it will be made of modules that are simple enough to understand, just like engineering is. I don't know. For me, that's a little boring. I don't want to be there. I'm interested in the principles. That's me, though. All right. We need to finish. Let's take a nice deep sigh of relief. Thank you for walking this path together. And I wish you a lot of excitement in your research, in your life, a lot of deep breaths. And thank you very much. So I applaud you. Bye-bye.